Hey y'all, my name is Prashanth Kumar, or PK, and I'm an MCAT tutor at Shemmassian Academic Consulting. Today, we're going to be doing a passage walkthrough in the MCAT Biochem section. You should be taking roughly 7.5 minutes on this passage, but today we're going to be taking our time, going more in depth, and seeing exactly what goes on in my head as I take on this passage. Let's get it. So I've gone ahead and put the passage on the left and all 5 questions are going through on the right. So feel free to take a moment, pause the video, and come back to us. If not, let's get right into it. First, let's talk strategy. For me, I want to maintain the same strategy regardless of what type of passage I have. This is important because it gives me something to stay grounded to, regardless of whether the passage is super easy or super challenging. I want to have the same structure in place so I'm not overwhelmed on test day. For me, this looks like reading a paragraph, writing a quick 3 to 5 word summary, then going on to the next paragraph, doing the same, on and on and on. The reason I like this is it provides accountability. It's sort of a mental check-in. Oh, do I know what's going on? Let me write 3 to 5 words to make sure, before I go on to the next paragraph and on and on. So think of that as a quick check-in, and it doesn't take too much time. The second thing on my strategy is I don't like to spend too much time on figures, especially on that first read. The reason for this is we don't know exactly what questions are going to ask about the figure, if any, and why waste time when time is such a finite resource on test day. So I usually just go through, maybe get a big picture idea of what the figure is looking at, and then go to the questions. Most of the times the question will tell you exactly what figure to go to, and you won't waste any time there, and we'll have to go back. So those are my 2 main strategies, and with that, let's get right into it. Triple-negative breast cancer, TNBC, is characterized by poor patient prognosis. TNBC cells lack expression for estrogen receptor, progesterone receptor, and human epidermal growth factor 2 receptor. The human cell line HCC1806 is used to model TNBC to study the disease. Okay, so the question I ask myself every single time is, what's happening? And that's a broad question that helps us hone in on exactly what 3 to 5 words we can use to describe this paragraph. So here it's saying we have this cell line HCC1806, and that has, I'm going to put a little X there, and I'm going to say no estrogen receptor, no progesterone receptor, and no HER2 receptor. Okay? And so remember, you're the only person who's going to look at these sort of abbreviations, summaries, symbols, whatever. So use whatever you need to. It doesn't have to make sense to anyone else other than yourself. So as you see, I just put little abbreviations, kept it really quick, and we're good. Let's go on to the next paragraph. Scientists have recently identified epigenetic dysregulation and chromatin remodeling as potential drivers for TNBC. Researchers hypothesized that a link between transcription and chromatin structure might provide clues into mechanisms behind TNBC. In HCC1806 cells, the researchers mapped genome-wide locations for RNA polymerase II, important histone variants, and 7 histone modifications. Okay, so again, we have a lot of details here. We're talking about different enzymes, different sort of structures, different things. But what is the big picture understanding? And this, I'm gonna write epigenetic mod to HCC1806. And that's like the big picture understanding. They're going to look for RNA polymerase II, histone variants, histone modifications, all of that. And if a question asks us, we can use that to come back to sort of where we are. But we don't need to spend too much time thinking about it before we need to. On to the next. The researchers used ProSeq to detect transcriptional responses to a variety of stimuli. ProSeq measures newly synthesized DNA, thereby providing a sensitive tool to detect rapid transcriptional responses instead of measuring only steady-state RNA abundance. In ProSeq, researchers isolate polymerase II bound to nascent RNA, add biotin to the RNA, digest the polymerase, and sequence the biotinylated RNA. Okay, again, what's happening, right? And so here we have this description of this technique. What's it called? It's called ProSeq, right? and then I'm just going to say technique, and then I'm also going to write plus measure new DNA. Right? Something quick, simple, nothing too crazy. That's the big idea, right? It gives us the step-by-step analysis of what exactly is going on in this technique, but we're just giving a sort of table of contents of what's going on there, big picture, and we can come back if we need to. Again, we have a figure, right? What did I say earlier? I'm not going to spend too much time on that figure on that first read, because inevitably, if we need to, we'll come back to it. But it looks like it's just giving us that sort of structure of what's happening for the pro-seq reaction. Final paragraph. After developing the method, researchers used pro-seq to measure the transcription of a putative oncogene after knocking out a related tumor suppressor. Okay, again, what's happening? And here, all I'm saying is X tumor suppressor, right? And then pro-seq oncogene. Okay, perfect, right? And with that, we've done it. We've done sort of our roadmap for our passage, and we're ready to go straight to the questions. So let's do that. All right. Question number 1. A researcher adds a protease inhibitor to the reaction mixture and finds that polymerase II bound RNA can no longer be isolated. The protease inhibitor A prevents digestion of the RNA, thereby not allowing the RNA to be dissociated from the protein. B prevents digestion of the protein, thereby not allowing the RNA to be dissociated from the protein. C increases the length of a sequencing runs. D prematurely releases the RNA from the protein. Okay, so this is a standalone question, right? It's not asking any question dealing specifically with our passage. And so with that, we don't even need to go back and we can think about what's happening, right? It's saying, okay, we add a protease inhibitor, okay? And find that polymerase II bound RNA no longer isolated, okay? So those are the two things. And so first, we have to note that we're not dealing with just a protease. It's a protease inhibitor, okay? And so what happens when we're inhibiting a protease? Well, then we can no longer find this RNA plus, I'm just going to write it as P2, polymerase II, okay? We're not going to have this sort of combination if we add this inhibitor. So what does a protease inhibitor do? That is what the question is asking, right? What is a protease inhibitor doing? And so to do that, we need to understand what is a protease, right? A protease is two parts. It's a protein nuclease, right? It's like an enzyme. Anything with an ase like breaks down, and the first part of it is what it breaks down. So ase is protein. So it's going to break down proteins, right? And so which of the answer choices talks about that? Well, A is talking about digestion of the RNA. B is talking about an inhibitor preventing digestion of the protein. So B is already looking like a great answer. C doesn't have anything to do with that. It's talking about the increasing length of sequencing run. And D doesn't really talk about prematurely releasing the RNA from the protein, right? We're talking about a protease, a protein like ase or destroyer, breaker down. And so B is our best answer. If we're having an inhibitor, right, we're going to inhibit breaking down proteins. So right away, we're 1 for 1. On to question number 2. What is the most likely effect of removing biotin from the experiment? A, the protein and RNA dissociate earlier. B, the RNA can no longer be pulled out of the reaction. C, the protein and RNA bind more tightly. Or D, the DNA is degraded before transcription occurs. All right, so when did we see anything talking about biotin? This requires us to go back to the passage, right? And so if we look through our passage, we're going to look at biotin and sort of this area right over here, right? It's talking about the step-by-step process of pro-seq. And we see biotin is mentioned right here. But let's just read that sentence all over again. In pro-seq, researchers isolate polymerase II bound to nascent RNA, add biotin to the RNA, digest the polymerase, and sequence the biotinylated RNA to what? To measure newly synthesized DNA, right? And so what is it going to do? What is this biotin doing? Well, it's a necessary sort of addition to help with this separation, right? What's the step before? Well, we're isolating polymerase 2 bound to RNA, right? So I'm just going to write out the steps right here. We have P2 bound to RNA. Then what happens? We have P2 bound to RNA. I'm going to write biotin, right? Because we have this RNA biotin. Next, what happens? Well, we digest the polymerase. So we get rid of that. So P2 goes away. So all we're left with is RNA biotin, right? And so then we sequence this. That's the step-by-step process if we wanted to write it out. So if we remove biotin from this reaction, we'd be affecting which step? We'd be affecting this step, right? The step where we add biotin. And as a result, what cannot happen, right? As a result, if we stop this, right, we put a little X there, that means we can't go further than this. And that means we can't get to RNA biotin alone in this sequencing. So which answer choice talks about that? The protein and RNA dissociate earlier? That's not necessarily true, right? We're stuck at this place. There's a little wall right here where we're stuck at this P2 RNA state without the biotin. So we can eliminate that. The RNA can no longer be pulled out of the reaction. Exactly, right? If we're stuck there, we can't get to this RNA biotin state where we can sequence it. So B is seeming like our best answer. C, the protein and RNA bind more tightly. It doesn't say anything about binding more tightly, right? Biotin didn't affect how strong the bond was, so we can eliminate C. D, the DNA is degraded before transcription occurs. Nowhere in this pro-seq reaction is biotin affecting DNA, so we can eliminate that answer, and right away, we're 2 for 2. Great work. All right, on to question number 3. 1, 2, 3. Researchers observe a certain histone post-translational modification in euchromatin. Which of the following do they most likely observe? A, histone 4 lysine 27 acetylation. B, histone 4 lysine 27 decetylation. C, histone 4 arginine 4 methylation. D, histone 4 arginine 8 methylation. All right, so here we have a question where we might be a little lost, right? I don't remember it talking about lysine or arginine or I saw histones in there. I saw maybe acetylation or modifications in there. But right, this is actually a standalone question. And they might be trying to trick you, right? There's numbers there. We have not seen these numbers anywhere in the passage. So we got to trust our instinct, understand that this is a standalone question, right? And what is the standalone question discussing, right? It says we have these modifications in euchromatin. Which of the following do they most likely observe? So to answer this question, we need to have an important understanding of the difference between euchromatin and heterochromatin, okay? And so euchromatin is open, okay? It's like more loosely bound, and heterochromatin is more tightly bound to the histone. Loose bound to histone. And so this is more openly transcribed, and this is less transcribed, because if it's tightly bound, it's going to be harder to access, right? So these are the most important parts of this. Okay, so with this, now we need to have a second piece of information. And that is what modifications are related to euchromatin and heterochromatin, okay? So right away, we have acetylation, deacetylation, and methylation in our answer choices. I would say we can already eliminate C and D because both of them are dealing with methylation, and so we should know that that's probably wrong. But methylation is associated with heterochromatin, or tightening up of this DNA. Acetylation is associated with euchromatin, so it loosens it up, okay? And by that vice versa logic, right, deacetylation would be associated with heterochromatin, and demethylation with euchromatin. So these are important sort of post-translational modifications of euchromatin that are very, very important. So I'm going to start acetylation, methylation, because those are the two sort of most important ones. And then you can get deacetylation, demethylation's effects from that. And so with this, which one is related to euchromatin? And that's acetylation, right? Not B, not C, not D, right? I said we could eliminate C and D already because both are dealing with methylation. B, also deacetylation, opposite of what we want. So right away, we used our piece of information that we learned from our content review to get this question. And we are 3 for 3. On to question number 4. What is most likely pro-seq result from the experiment the researchers conduct in the final paragraph? A, tumor suppressor protein expression is increased. B, tumor suppressor protein expression is unchanged. C, oncogene transcription increases. Or D, oncogene transcription decreases. All right, so tells us exactly where we need to go, right? Tells us we need to go to this final paragraph. And so what does it say? After developing a method, researchers use pro-seq to measure the transcription of a putative oncogene after knocking out the related tumor suppressor, right? And so we can use these sort of words to help us think about what's happening, right? So what would a tumor suppressor do to an oncogene, right? A tumor suppressor does what to an oncogene? It suppresses this oncogene. Okay? And so it says in this final paragraph, we're running an experiment on a knockout of a related tumor suppressor. And so we're like getting rid of this. So I put an X there, or you can put a little down arrow. And I like to think about what happens to the oncogene if we have less of that, right? Well, if we have less inhibition, that means oncogene increases, right? Sort of think about how the arrows follow, right? We have a downward arrow and then sort of inverts it with this inhibition. So we're going to have increased oncogene. And so right away, we see one answer choice that stands out. Oncogene transcription increases, right? If we have less sort of transcription of this tumor suppressor gene that is inhibiting oncogene, it makes sense that that oncogene expression increases. So right away, very solid question number 4, and we're all to question 5. Researchers add estrogen to a culture of HCC 1806 cells. A control culture of HCC 1806 receives DMSO. Pro-seq data most likely indicates what? A, an increase in estrogen-related signaling pathways in the estrogen-treated HCC 1806 cells. B, similar new gene transcription profiles for the two cultures. C, an upregulation of newly synthesized estrogen receptor in the estrogen-treated HCC 1806 cells. Or D, a downregulation of newly synthesized estrogen receptor in the estrogen-retreated HCC 1806 cells. Again, a lot of details right here, right? We never really talked about DMSO or like a lot of estrogen, but there was one part of our passage where we did talk about estrogen. And where was that? Very, very early on, right? That was right here. And we have it in our sort of summary right here. TNBC cells, or in this case our HCC 1806 cells, lack expression for estrogen receptors. And so that is very, very important, right? If we lack expressions for the receptors, so no receptors, that means nothing can bind to it, right? Nothing can bind, estrogen cannot bind to our receptors if there's no estrogen receptors, right? So that we're gonna put a little X there, and that means regardless of whether we add estrogen to it, nothing can happen. That's the whole point, right? If we don't have receptors, we can't bind the thing. And so right away we should know nothing should happen to our first culture of HCC 1806 cells. And so right away we can eliminate A. B, see it says similar nutrient transcription profiles for the two cultures, so we can keep that, right? An upregulation of newly synthesized estrogen receptor in the estrogen-treated HCC 1806 cells, cross that out. A downregulation of newly synthesized estrogen receptor in the estrogen-treated HCC 1806 cells, D, we can eliminate that, right? The whole point is these cells don't have receptors, so they're not going to react to any increase or decrease in estrogen. So we can eliminate A, C, and D, and we're left with B. We also see that this second part of this question, right, DMSO. DMSO is a commonly used solvent. I wouldn't even worry about it too much, but think about if you add just a random solvent that isn't going to affect a reaction too much, nothing else would be affected in the reaction. So that's why it's saying similar new gene transcription profiles for both the cultures, right? If we're not going to have an estrogen receptor, estrogen's not going to affect it. And if you add DMSO, a sort of solvent that doesn't really do anything, nothing is going to change in this pro-seq analysis. And there we are. We're 5 for 5. Great work. Great work, team. We crushed another biochem passage. Hopefully that was super helpful and you got some practice sort of breaking down experiments. Pro-seq was very big in this one, and you understood exactly what each step was doing and how to sort of draw that out for yourself. If you like this video, give us a like and subscribe to our channel down below, and comment exactly what you want to see next so we can help cater these for you. Additionally, go check out Vikram's 528 strategy series, a super helpful series to help you master your MCAT strategies. Additionally, sign up for our MCAT question of the day down in our bio so you don't miss out on any practice before your test day. Happy studying, and let's get it.